Globally, women outlive men, and scientists have mostly connected these sex-based disparities in longevity to biological factors that contribute to survival. But you don't have to think that only human males suffer. There are significant disparities in life expectancy and aging among different mammalian species, according to a recent study of wild mammals. Despite the fact that females often outlive males across all species, mortality risk does not rise as quickly in males as it does in females. Consequently, they assert, there must be additional, more complicated elements at work, such as the animal's living environments and sex-specific development, survival, and reproduction over the history of the species. It is to see that this gender gap in longevity frequently exceeds the gender gap in lifespan seen in humans, while also being very varied among species. In humans, women typically have a life expectancy that is 8% longer than men's. But among wild animals, females live 18.6% longer on average than males. For several groups of mammals, the ratio varies greatly. Additionally, a woman's extra years are usually healthy ones. American males can anticipate living 67 healthy years, while American women will experience 70 years of full health, according to the World Health Organization's HAG-AL Index, which determines how long a person can expect to live without developing a serious illness or injury. With impressive life extensions achieved upon either dietary restriction or lowering activity of insulin/insulin-like growth factor 1 signaling and target of rapamycin and nutrient-sensing pathways, research on laboratory model organisms has revealed a pivotal role for diet and metabolism in determining lifespan. But it's interesting to note that different sexes respond differently to mutations that impact these pathways, with females experiencing a larger life extension and males experiencing a muted or non-existent response. Studies have shown that sex hormones have a significant impact on aging processes. Data on castrated men, for instance, suggests that testosterone causes aging to occur more quickly. Growth and development, metabolism, and behavior are all significantly regulated by steroid hormones, including sex hormones. It's probable that the interaction between sex hormones and nutrient-sensing pathways leads to varied lifespan and health outcomes for the sexes. One of a pair of chromosomes that determines an individual's genetic sex in mammals, some insects, and some plants is the Y chromosome. Scientists were able to verify the idea that chromosomes determine the sex of developing creatures in the 19th and 20th centuries, thanks to the development of new microscopic and molecular tools, including DNA sequencing. The Y gene's chromosomes aid in the development of the sperm cell, which is the male gamete in an adult organism. Lionesses, for instance, live in the wild at least 50% longer than male lions. The researchers collected information on lifespan and sex chromosomes from 229 different animal species, ranging from insects to fish and mammals. Animals with two of the same sex chromosomes typically lived 17.6% longer on average than those with two distinct sex chromosomes, according to data published in the Journal on Biology. It's interesting to note that in species of birds, butterflies, and moths, when the male shares two identical chromosomes, this pattern persists, which is the opposite of mammals. This occurred due to the Y chromosome's lower size than the X chromosome, which prevents it from being able to hide an X chromosome that contains dangerous mutations. The gender with one shorter sex chromosome persisted longer than the gender with two longer sex chromosomes. Males who are free to roam may be more vulnerable to environmental infections. 3 populations of bighorn sheep were affected by this. According to the study, the deletion of the male sex chromosome results in cardiac muscle scarring and can cause fatal heart failure as men age. A specific kind of chromosome that plays a role in determining sex is called a sex chromosome. The 2 sex chromosomes, X and Y, which are present in humans and the majority of other mammals, work together to determine a person's sex. Males have 1 X and 1 Y chromosome, while females have 2 X chromosomes in each cell. Despite the fact that these 2 chromosomes join with one another during meiosis, there is typically very little homology or recombination between them, mostly due to a significant disparity in their genetic makeup and size. Often, one chromosome is smaller and seems to contain solely the genes required to determine sex. The XUE system has two X chromosomes in females and one X and one Y in males. Males are hence referred to as heterogametic, since they can produce either X or Y chromosomally heterozygous gametes. All of a female's eggs possess one X chromosome since she is homogametic. Humans and other primates use the XUE sex determination mechanism. The study is the most recent to examine the issue known as mosaic loss of Y, which occurs when the Y chromosome vanishes from a part of a man's blood cells. Although the cause is unknown, aging is thought to be a contributing factor. More than half of people who live into their 90s and 40% of men in their 70s are expected to have it. A short, stumpy chromosome, known as Y, was once assumed to be lost as a natural component of aging by biologists. The Y chromosome is commonly thought of as the sex chromosome, since men have both an X and a Y, while women have two X. Males who display Y chromosome deletion have been reported to die earlier and have age-related illnesses like cardiovascular disease and Alzheimer's, which may help to explain why men often outlive women. Blood cells and other cells that experience fast turnover are particularly susceptible to Y chromosome deletion. The trait is not genetically inherited, though, because Y chromosome deletion does not happen in male reproductive cells. Previously, scientists believed that the Y chromosome mostly determined the traits associated with male sex. The Y chromosome, however, contains more genes than was previously believed, many of whose functions are still unknown, according to studies from recent years. Research has connected Y loss to a number of illness risks concurrently with that work. Local environmental factors that compromise between reproduction and survival may also have an impact on the size of the lifespan disparity. Males in some animals devote more energy to sexual competition and reproduction, which, according to scientists, could result in greater lifespan disparities between the sexes. The fact that males are more likely to survive when they offer some or all of the parental care may also account for the sex gap. Females incur a high health expense when they give birth and raise children. This expense is minimized if both parents cooperate in raising their children. The FDA has already approved the drug pirfenidone for the treatment of idiopathic pulmonary fibrosis, a kind of lung scarring. The current study also found that men who exhibit a Y chromosomal deletion may benefit from treatment with this medication. Antifibrotic medicines are currently available and are used to treat lung scarring. One concern is whether or not males with Y loss would react to those medications particularly effectively. What causes Y loss is one of the unsolved problems. There is a ton of variation. At age 60, some men exhibit significant Y loss while others exhibit relatively minor Y loss. Women don't have a Y chromosome, but as they age, one of their X chromosomes can go from their blood cells. Walsh noted that it is less typical than Y loss in men and that there is currently no evidence connecting it to a specific illness risk. Reservation suggests that one reason why women typically outlast men is due to the loss of Y. Younger males passing away from accidents and other causes can partially account for that gap, but it's not the whole explanation. Watch thoroughly and you will see how sex steroids like androgens and oestrogens control biological functions that may affect aging and longevity in order to understand how sexual dimorphisms in vertebrates are mediated. With females displaying greater plasticity for life extension, the sexes can react to food restriction and changed activation of nutrient-sensing pathways differently. The master regulators of sex differences in mammals are androgens and oestrogens. They have an impact on a variety of organismal, cellular, and biochemical processes, which in turn regulate a variety of life history traits. There is evidence that sex steroid hormones have a drastically different effect on lifespan in men and women. Whereas testosterone is removed by castration, oestrogen appears to play a number of protective and advantageous roles. Whether it's whales or people, on average, male mammals die younger than female ones. For some reason, the men pass away first. This is frequently attributed to sexual selection. Males typically have to compete more fiercely for a partner, such as when stags lock antlers, and this seems to wear them out.